Morning, everyone. Aksa Xeneza from the University of Rwanda. I'm an assistant. I'm very glad to join this fifth online sensitization program for strengthening fundamental research capacity with Drosophila in Rwanda on this day of 21st August 2024. This online sensitization was prepared in a collaboration of INSTAL Africa, which has a collaboration between FILAT, Rwanda, Drosophila, NCST, RBC, NIRDA, UR, FDA, and this INSTAL Africa has started since October 2023. So as some of you knows, there are other 4 online sensitization that has already ended, and now we are going to have this 5th one that will have the main speaker, Dr. Hellen from Aga Khan University, who will be sharing with us the Drosophila melanogaster solution for research in low resource settings. Without taking much time, I'd like to thank everyone for joining us. And let me invite, before that, let me just introduce to you and welcome our special guest of honors, Professor Abraham Mitike, the Principal of College of Medicine and Health Science from University of Rwanda, Dr. Eugene Mutimura, the Executive Secretary of NCST, Professor Claude Muvunyi from RBC, the DG, and the DG of Institute of Research and Development, Dr. Sekomo, and Professor Emile Bienvenu, the DG of Rwanda FDA, also Professor Leo Mutesa, the Professor of Human Genetics, College of Medicine and Health Science. And also, I welcome everyone, one online who are following us. You are all welcomed to interact in these sessions. And without taking so much time, I'd like to welcome Benjamin from NCST to give us a brief introduction of our special guest speaker, who is talking to us. Thank you very much, Benjamin. The floor is yours.
Thank you, Ijezeneza, for such a wonderful introduction. Good morning, everyone. My name is Benjamin Nabana. I work at National Council for Science and Technology as STI Strategic Partnership Specialist. And today I have the honor to introduce our special guest speaker, Dr. Hellen Kinyi. Let me give the profile of Dr. Hellen Kinyi. Dr. Hellen Kinyi is a seasoned medical biochemist with a robust background in clinical medicine and community health. Over the past 15 years, she has dedicated herself to academic field, teaching medical biochemistry at various prestigious institutions, including Kampala International University, Clark University, and Kabale University in Uganda. Currently, she is a faculty member in the Department of Biomedical Sciences at the Medical College of Aga Khan University in Nairobi. Dr. Kinyi's journey with Drosophila melanogaster as a research model began in July 2013, when she participated in a course organized by Drosophila titled Drosophila in Biomedical Research, Affordable and Impacting, at the Institute of Biomedical Research (IBR) of Kampala International University. This course provided her with the foundational knowledge necessary to employ Drosophila in biomedical research. Her expertise in this area was further enhanced between August and September 2013, when she attended the Second International Brain Research Organization (IBRO) School on Insect Neuroscience and Drosophila Neurogenetics, also held at IBR. These advanced trainings equipped her with specialized skills in using Drosophila melanogaster for research, particularly in the fields of neuroscience and neurogenetics. Throughout her career, Dr. Kinyi has successfully applied the Drosophila melanogaster model to both her research and teaching. She has trained her students in areas such as antioxidants and nutritional biochemistry, contributing significantly to, to the understanding of antioxidant activity in locally consumed vegetables and impact of micronutrient ratios of health and diseases. Her current research focuses on exploring how varied dietary macronutrient ratios can modulate the immune response, further advancing knowledge in the field of nutritional biochemistry. Finally, her dedication to research and education continues to make a meaningful impact, fostering a deeper understanding of biomedical science through the innovative use of Drosophila melanogaster as a model organism. Please welcome uh, Dr. Hellen Chinyiji over to give her her talking.
Thank you, Benjamin, for the great introduction of our main speaker today. As Dr. Helen is uploading the slide, allow me to acknowledge the presence of another special guest of honor. We are very thankful for Dr. Olaleye, Director of Lagos State Polytechnic from Lagos, Nigeria. We are very grateful for you to join us. Thank you very much. Dr. Ellen, the floor is yours. Thank you.
Thank you so much. Thank you, Benjamin, for that introduction. Um, when I was given this topic to discuss, I thought to myself, how do I approach it? So I'm going to take you through a journey of my growth as a researcher. And I hope as we discuss how I've reached a tiny bit where I am, we can be able to understand how research in Africa and the challenges we find in Africa, and how Drosophila melanogaster would play an important role not only in teaching but also in helping us teach our students. So this I will make this talk very interactive. It will be nice to hear your point of view also, and so I welcome you to this discussion. And I will be welcoming questions even as we continue. So this is the outline that we're going to use. We're just going to see how research was before Drosophila, then the introduction of Drosophila, and some of the experiments we've been able to conduct using Drosophila melanogaster. I hope you can all hear me and you can see my slides well.
Excuse me, the slide is not moving. I don't know.
Slide is not moving. Okay.
Maybe, yeah. Okay, now we are on the outline
Okay.
slide. Maybe if you can put it in presentation mode, if you don't mind.
Yes, this is the presentation mode.
Oh, okay.
It's moving now?
It is. We are on outline slide, the slide of outline.
Yes. Yes.
Oh, okay. Yes, we can see it. Thank you.
Okay. I guess I'll be going back and forth because it seems to move. I need to move here. Thank you so much for that feedback. Before I continue, I would like us to go to mentimeter.com. We're going to use Menti as our interactive platform, and kindly request you to, in less than 10 words, maybe mention what your expectation from this session. What are your expectations from this session? So you can scan this QR code to get into Menti, or you can go to the website menti.com and use the code 39042865 to log in, and maybe type in less than 10 words, what do you expect from this particular session? So are we there? Are we able to access the code? And as you
Yes,
open this,
yes.
yes, as you open this, don't close it. We'll be using it again and again to interact with the presentation. Thank you so much. You can see there's one response to the importance of Drosophila melanogaster in diagnosing some diseases from low and middle income countries. Thank you to learn about melanogaster. Yeah, I believe you will learn one or two things about melanogaster. We're going to see how we can use it in diagnosing and also managing some as using it to assess possible therapies to some of the diseases in low and middle income countries. Can we give it a few more? Someone still typing? Thank you. To gain more knowledge on Drosophila husbandry and research and potentials in a low income center. Yes, we're going to see some of this, maybe not so much on the husbandry, but some of the experiments and some of the work you can do easily with Drosophila in our labs that we find in our institutions. So thank you so much. As we start our journey, I want to start with just talking about some of the work I did before Drosophila and to give you some of an idea of some of the thinking that went behind this work. For my undergraduate, my first degree, BSc Biochemistry, we were interested in a plant referred to as Ficus natalensis. It's the fig tree. And in Uganda, it had been used commonly for ulcers, for management of stomach ulcers. And so we had the idea initially to assess its effect and its anti-ulcer effects. So me and my supervisor, we worked on a proposal and we had a very elaborate work from assess its extraction through the various methods all the way to giving it to an animal model. At that time, the animal model was the rat that was there at Kampala International University. However, when it came to the point of feasibility of the study, the experiments were very expensive. The number of rats that are going to be used, the reagents that were going to be used. And I remember in my presentation when I was defending my proposal, the head of department was asking me, but who is going to fund this research? Again, I was a privately sponsored student, actually on a staff development program. The university was paying my school fees, but was not able to fund my research. And he was very worried, how are you going to fund this work? And again, we had to rethink our strategy. And so we came up with an idea. He gave us the idea that we have the animals in the lab. We have these kits that we use to teach undergraduate medical students. Or why don't we do something within this area? And so with that in mind, my work was reduced to assess the liver toxicity of the crude extract of Ficus natalensis. Yes, we did some research. We were able to see how the extract affected the body weight of the animals. And we can see we extracted the Ficus natalensis. We saw how, as the increasing doses of the extract affected the body weight of the animals over time. We saw that the extract was able to cause the liver to enlarge. And using the kits that were available, we were able to see its effects on liver function, affecting the AST, ALT, and serum GGT. However, remember, this is not what I really wanted to do. But due to limitation of funds, limitations of resources, I was limited to this. So well and good, I was able to graduate and move to the next level, and we even got a paper published. However, there was that feeling of this is not adequate, I could have done better work. So again, for my master's level, I again my idea now, remember I came from a clinical background, And I had worked a lot with women and children in obstetrics and gynecology. And I had the burden on how can I link my clinical background with this biochemistry. And so my idea was how can I link? And I had the idea to measure the amount of nutrients that are there in women who are pregnant. So I wanted to do approximate study of the foods. However, again, the same challenge came back again. Again, I was a staff in a staff development program. I was not funded for my research. We could not find any funding at the moment. And now I had to think back at what can I do? What can I do? Which work can I do that can allow me to get an MSc? Okay. And being biased with nutritional biochemistry, I had to look for a topic within nutrition. So I ended up assessing how the cooking method of vitamin C affects the antioxidant activity. But you can bear in mind this was not really what I wanted to do. But even within this study, there were several methods I could not be able to do. There were reagents like DPPH that were very expensive. Again, checking how this antioxidant activity was going to happen in vitro was a bit of a challenge, especially thinking about the cost of rats at that time. So I ended up again just extracting vitamin C, assessing how the vegetables, when they were raw and cooked, and measuring the vitamin C levels, measuring the antihemolytic activity in these vegetables. And we were able to also get another paper. We used reducing power activity. Again, this was using reagents that were readily available in the lab. For example, for reducing power activity, we were able to use ferric oxide that you gotten that was available in the lab that is easily used in biuret reagent or Barfoed reagent that we use for qualitative analysis. And so we use this reagent to be able to see how the antioxidants in the vegetables were able to convert iron 2 to iron 3. And this is what we measured the difference in absorption, and we were able to come up with the reducing power. Again, hydrogen peroxide scavenging activity. We know hydrogen peroxide is a common reagent in the labs, and we were able to use this. However, even though we're able to do experiments and get papers published, there was that feeling that I could have done more, I could have done better. But resources, some of the equipment were not available. So this is what I was able to do before, as both as an undergraduate and a postgraduate student. We were able to get some of the work done, but again, we had challenges. So from this small talk, this small part, I'd like to ask, so what factors came out to you in my discussion that could have influenced my choice of research topic in the above two scenarios? What factors were you able to notice, or what factors came out to you? Just using the same codes, you can go to menti.com 3904-2865. What are some of the factors that influence the kind of researches that are done based on my stories or even based on your own experience? So what factors influence you? What research topic you're going to choose? Lack of adequate funding. So this is usually a very big problem for many of us, especially in privately sponsored universities or even in general universities. We lack adequate funding, and so it limits us in what we can do. Yes, reagents also are a common problem. The cost, yes. So many times you have to buy our animals, you have to look for reagents, you read about elegant experiments, and when you go down to look at the cost of maybe a reagent that was used, maybe it's not commonly used in the lab. Like for me, DPPH was a very expensive reagent. I remember being told like 5 grams of DPPH
Hello.
Hello, I think maybe it's a network. I can also not hear from my end, yeah?
Okay. I hope we can wait for a few minutes and while the speaker is working on the network, I guess it is a network issue.
Hello, can somebody hear me?
Yes, now.
Now you can hear
Yes,
me?
yes, yes.
Yes. Okay, thank you. I just changed my Wi-Fi. I'm at work. I'm using a Wi-Fi. I think the one I had was not working. Thank you for that information. I could have gone very far away. Where did I leave you people?
Actually, at the Menti, when people are responding on the drive for the research topic for,
For the mentee.
yes,
Okay. So I think from here I just started talking about we have limited reagents. And yeah, we saw some of the challenges that are common to all of us in Africa in conducting research, mainly in terms of lack of adequate funding and limited access to reagents. Okay. So in 2013, as I was doing part of my master's work, Dr. Mata had joined KIU after a session with with Trend in Africa, and she had established a lab called Institute of Biomedical Research. And so part of my work was done in this institute. And when she talked about Drosophila and starting a course with Dros Africa, I had no option because I was already in the lab. I joined the course. So I was explaining that one of the things that really influenced me in this course was bringing into life some of the concepts we had learned that were not realistic. For example, when we learned about molecular biology, DNA transcription, translation, we thought about the things that Watson and Crick did somewhere far away. But here was a fly that was brought that they had modified the gene expression using the UAS Gulf 4, and suddenly all these concepts that you were taught that were theoretical were actually here. They were actually here with us. And we learned a lot during this. And Drosophila was introduced to us as a possible model for research. Not only was it as a possible model, but it was also a cheap model and allowed us to be able to do research with some of the factors that were hindering us, especially in terms of rats. The Drosophila first thing has a very short life cycle of up to 5 days. In 5 days, we have the females laying their eggs. They go through the life cycle of larva, pupa, and in 5 days they have hatched. We have adults, and in 5 days these adults are ready to mate again and reproduce. So some of the challenges we had about using mice, the N for making experiments that were reproducible, statistical significant results, could be addressed by Drosophila. Now, the other thing, especially with me from a medical background, was really the fly and humans, how can I take my results that were from flies and relate them to human? And it was a very big surprise for me to realize we have as much as 60% conserved genes between us and the fly. And so as many as 75% human diseases could be modeled in the fly. And so this made it possible to use Drosophila for several, especially neurodegenerative diseases like Parkinson's, Huntington's disease, and Alzheimer's disease. And at that time, there was a lot of research happening in aging using Drosophila melanogaster, immunity, diabetes, and cancer. So this was really groundbreaking and shaking all that we had been taught for me in my undergraduate and postgraduate about doing research. Around the same time, around in July, Trend came back for the second IBRO School of Insect Neuroscience and Drosophila Neurogenetics. The first time I was very hesitant to attend because I thought insect was very far away from me as a researcher with a medical background. But this time, after hearing some of the concepts with Drosophila Africa, I jumped right into the class. And Trend worked with IBRO. Excuse. Hello, can you hear me?
Yes, we can. Yes, please.
You are hearing me. Okay. I just got someone telling me they cannot hear me.
Yes, the voice just came back now.
Oh,
Mm.
I don't know what is happening to my Wi-Fi. It keeps on going back and forth. Okay.
I think it's fine now.
Okay, please, when I get lost, let me know so that I don't go very far ahead.
Okay.
I was talking about the second IBRO School of Neuroscience that was brought back with Trend. And the second IBRO School, for me, it was like now reinforcing the concepts I had had. With Drosophila Africa, some of the concepts were very new, and at some point they did not really make sense. But at this time, when I did them again with Trend, the concept were now like reinforcing what I had learned. I learned how Drosophila can be used to model human diseases like Parkinson's and Alzheimer's. I learned how they brought with them flies that had been genetically modified. For example, the pink fly was one of the flies we were introduced to. And this fly was more sensitive to oxidative stress because it had been modified. Some of the enzymes that could be protecting against oxidative stress had been downregulated. And so it was more sensitive to oxidative stress, and it was a very good model to study oxidative stress. We learned about bank sensitive flies that were very good to study epilepsy, because epilepsy was one of the areas of interest. We further learned how to separate male and female flies. And something that really up to today made a very big impact was introduction of Mendeley as a referencing system. Truthfully, before I had not heard about Mendeley or any other referencing system. People had been talking about EndNotes, but in this insect neuroscience, in IBRO school, we had a special session on Mendeley, and up to today, as a PhD and in my researches, and even for my students, I still use Mendeley as a referencing system. So the trend workshop was long. It was 3 weeks of this workshop, and not only did they give us opportunities to study, they also made us learn things practically. We made our own kits to be able to study science, to measure electrical activity in the muscle of insects. We did the antioxidant assays ourselves. We introduced hydrogen peroxide and observed how the flies behaved between a control fly, white, and the pink fly. We read about Alzheimer's and did experiments with flies that were models for Alzheimer's. We had a wonderful time. So after Drosophila, some of the works that we presented, these were very simple experiments that we did. We can see we had the normal fly white and the pink mutant, and we exposed it to hydrogen peroxide for 24 hours, and we could see the difference in survival. Most of the flies that were sensitive to oxidative stress, the pink fly died. We also exposed them to osmotic stress using ammonium chloride. And again, we saw the pink mutant flies were more sensitive to oxidative stress, as they were showed by lower survival compared to the white fly that was not did not have any mutation in relation to oxidative stress. Other things that we're able to assess was heat stress, and this was a very simple experiment where we exposed, as you can see in that diagram, we had the flies in a container, in a beaker with water, and we increased the temperature. And we saw, despite the pink fly being more sensitive to oxidative stress, it was more resistant to heat stress. You can see as we increased the temperature, more of the white flies that were resistant to oxidative stress were knocked down, not actually died, but were knocked down faster than the pink mutant fly, while of this knockdown, some of the white flies did not recover. Yet the pink mutant fly that was more sensitive to oxidative stress was more resistant to heat stress. So these experiments were really mind-blowing, very simple experiments. All we needed was the fly, a water bath, and a thermometer, and you could actually come out with results. You can see here, Jorge, one of my mentors during the trend, actually made a slide and actually showed us this can be done. And it was a mind-changing for me because, this simple experiment with the fly can actually make a paper or can make slides that can be presented in a respectable way. So this was really, really mind-blowing for me. And it really created a big brain shift for me in terms of research. So from this session, I'd like to have a small question. Which of the following is true of Drosophila as a research model? You can go back to Menti. There are a few options. Which of the options is true of Drosophila as a research model? I hope you're with me and I've not lost you.
Yes, yes.
Yes, thank you. So someone says 80%, there's 80% conserved genetic sequences with humans. Can we hear more responses? Do you all agree with this one as the correct one? 3 say 80% conserved genetic sequences. 4 give that same responses. Anyone with a different answer? Five. Okay. 6. Go for this.
Sorry, from the YouTube end, people are commenting that the slides are not moving.
Oh, but you guys are seeing my slides move. Okay, I will keep on shifting back and forth. Okay, so we have 2 saying the females have high fecundity, 8 saying 80% conserved sequences. We give it 1 more minute to see the responses. At least we all agree the fly does not have a short life cycle of 20 days. Yeah, we saw the life cycle is very short of 5 days. So clearly that is not right. Most of us are saying 80% conserved genetic sequences. However, that is not correct. Only 60%. We have 60% conserved genetic sequences. However, the females have very high fecundity. That is the correct answer. A female in one session can lay as many as 100 eggs. and in its lifetime can lay as many as 2,000 eggs. So the answer is actually option 2. Females have high fecundity. We only have 60% conserved sequences. However, as many as 75% of human diseases can be modeled in Drosophila. I think that's where we have the confusion. So thank you so much for this session. So our answer is actually 2, not 3. So now we move to the next phase, post-Drosophila. So now my mind has been open. Possibilities of research with this fly are possible. And we have a lab, Institute of Biomedical Research. And now I'm excited that I can actually move to the next level and use an in vitro model, an in vivo model. And so we started doing some work, of course, with Dr. Mata to move my master's research to the next level. And we had in mind to introduce the antioxidants that we had seen in the in vitro assay with the leafy vegetables into the animals. And we thought of using the wild type and the stress sensitive, the pink fly, to introduce the antioxidants and see how safe they will be in the flies. And one of the essays we wanted to do was total reducing power. Remember, the reducing power was using iron, so it was possible to use it in both in vivo and in vitro experiment. But now we had to think about in vivo antioxidants. So we thought about the superoxide dismutase assay and the catalase assays, which were superoxide dismutase and catalase enzymes are in vivo enzymes that are present in the fly. And I'm happy to say this work was actually sponsored by Sciency, and we acknowledged this paper was submitted to FASEP, and I think it has been cited twice or thrice. And we acknowledged, of course, the support of Africa and Trend. So during this time, I started working in how can I create a superoxide dismutase assay. There was one paper that was written in 1971 by Mivorich, and they described how adrenaline undergoes an oxidation in the environment. And so I thought we can actually try this out in Drosophila. And the experiment was very simple. All it required was a spectrometer, which was available, epinephrine as a source. We know this is available, which is given IV in terms of cardiac arrest or things like that. And sodium carbonate is a reagent that is commonly used. And all you had to do was get 5 flies, crush them, and obtain a tissue homogenate. So we did try, and we did actually find we can actually measure superoxide dismutase activity in the flies. In this first slide, you can see we can see adrenaline autooxidation. This is how adrenaline autooxidizes in the absence of any inhibitor. Okay, so we can see the absorbance at for 80 nanometer. It also, as more adrenochrome is formed, we have the superoxide being formed, and this is what is responsible for the increase in absorption at 480 nanometer. So to see if the superoxide dismutase enzyme in the fly was able to inhibit this activity, we did the same experiment with adrenaline, but now we added the tissue homogenate. And we can see there's a clear difference in the curve of absorption. We can see here the range was 0.02, 0.04, but you can see now the range is a bit lower. So the rate of autooxidation has reduced, and we can attribute this to the superoxide dismutase in the enzyme in the fly homogenate. And we're able to see that in this work, as much as we have 5 flies could cause as much as 12.5% inhibition of the autooxidation of adrenaline. And so this was very exciting because now we found a way that we could actually use the tissue homogenate. We could be able to measure the antioxidant activity of superoxide dismutase using the fly homogenate. Again, this was a very simple experiment. So these were some of the slides I was presenting in the lab meetings at IBR. Around the same time, I was interested in fluoride. I had my son, and I was worried about the amount of fluoride in water, just like any other mother. And there was this conversation that was happening, I think, worldwide about fluoride and cognitive function. And fluoride was there in the toothpaste, and everyone was saying we should not use fluoride. So I was allocated two students. Now they are doctors, Dr. Patrick Oga and Dr. Steve Odomel. They were pharmacy students. And I was curious, like, what is happening in our environment? So Patrick was able to measure the fluoride level of the water that we get from the tap. And that time there was water shortage where KIU was, and so we were able to go to the springs around and to measure the fluoride level. But with Steve, he used Drosophila, and he used Drosophila to assess what are the effects of fluoride levels in learning and memory. So in this case, it was very interesting because Drosophila now, the number of animals was not a problem. So we were able to create 5 repeats of the experiment, and each cycle of the experiment had 10 flies. And we introduced fluoride in the food of Drosophila at doses that were equivalent to very high fluoride levels, which had been classified by World Health Organization as being high. So we can see we use doses of 1.5 milligram per meal, 3.0 milligram per meal, and 5 milligram per meal. These are very high fluoride levels. You will relate them to human doses. And we assess them, how would they affect the short-term memory in flies. So what we did, we used a modified T-maze. Again, this picture, I got it from Africa. Trend in Africa. This is a picture. I don't have the actual picture, but we created something similar like this. We used some of the containers. We created a T-maze. And in this T-maze, we were able to put the flies at the bottom aspect of the T-maze and put it in a dark area. And we were able to train the flies by connecting it. In one aspect of the T-maze, we put acetone, which smells badly for the flies. And in the other area, we did not put anything. And we linked this acetone smell to a red light. So we're able to create a contraption where we're able to expose red light in one corner. We know Drosophila is attracted to red light, but we associated the red light with the bad smell of acetone. And so every time Drosophila tried to go this way, they'd find the bad smell and they would try to avoid that area. We did this experiment over 5 seconds, over 5 sessions to help them learn. And then we let the Drosophila rest for 20 minutes, and then we redid the experiment. And we tried to learn how they were able to assess their short-term memory. And we could see we did this to flies that were exposed to this food that had high concentration of fluoride for 7 days, for 14 days, and 21 days. And this very simple experiment showed us impressive results. If we look at the control animals, we can see how learning and memory declined with age. Remember, the control animals had a normal diet, but were not exposed to any fluoride in their diet. We didn't add any fluoride, any extra fluoride, because we even used distilled water to make their food, to make sure there was no fluoride introduced into their diet. So we can see as days progressed, animals fed on the normal diet, their learning and memory declined. The Drosophila live for an average of 28 to 30 days. So in 21 days, they are very old flies. But also of interest is as the concentration of fluoride increased in the... Hello?
Hello,
Yes,
yeah.
You're with me, yeah?
No,
Okay.
the slides are not moving. They are
I'm
not
not moving
moving.
the slide. I'm still in the
now,
slide.
Okay.
Yeah, I'm
now it
just...
is showing the 24th slide.
Yes, I'm showing the 24th slide.
It
On
was
the
not
YouTube
moving.
end, it's not moving. It's on the slide 7th.
Oh my, how can I control the slides on YouTube?
But don't worry about the slides on YouTube. It is moving as you are moving it. Can continue.
You can continue now. I think he has adjusted it.
Yes, I've
Okay,
adjusted it.
thank you
Okay.
so much. Thank you. Okay, so we could see as the concentration of fluoride was increasing, the learning and memory of the Drosophila was declining. And even though it does decline with age, the older the flies get, it was much more in flies that were exposed to high fluoride. So this was a very simple experiment that gave us very great insights in the role of fluoride in learning and memory. We also assessed using this very simple contraption. You can see this is a measuring cylinder. And in the measuring cylinder, we know the flies have what we refer to as negative geotaxis. When they are placed at the bottom of a cylinder, they will tend to fly to the top. So using this cylinder, we're able to assess how in a particular period of time, in this experiment we used 30 seconds, how many of the flies had moved to the top of the measuring cylinder. And we classified them after 30 seconds, how many flies were under 4 centimeters, how many flies were between 4 to 8 centimeters, and how many were over the 8 centimeter mark. So this experiment is very good because when the fly flies up, you're able to assess its locomotor, its muscular function, not only the muscle, but also the nerves that control the muscles to allow it to fly. So it's a very good assessment of locomotor function. And again, similar, we saw decline in locomotor function with time, which is expected with age. But also of interest, we could see, if you look at the green bar shows the flies that were over 8 centimeters. It was very interesting to see the flies that were exposed to 1.5 milligrams per meal per liter. At the beginning, many of them flew up. They had very high locomotor function. But as the dose of fluoride increased, the locomotor function declined. With time, we can see this number kept on declining.
You
Fewer
can
and
check,
fewer of the Yes?
still the slide is not moving. Maybe you can be using is it the left side to change the slide?
Okay.
You can click on slide 15. 25th, I mean. Yes.
I'm on slide 25.
Yes, you can be
moving.
using this. It is moving now. You can maybe use that same
I had not
technique.
moved my slides at all. I
It
was
is
just
on the
describing
25th.
that slide.
We're not seeing it.
Okay. Okay. So the locomotor function also was able to be assessed, and we could be able to see how fluoride affects the locomotor function. So around the same time as Patrick and Steve were in the lab, there was also another students, Sarah and Oliver, who are also pharmacy students, but were being supervised by Kenneth. And they were studying monosodium glutamate. We know monosodium glutamate is an ingredient that is put in some of the additives that we add in food, like Royco. And there was again a similar narrative about possible toxicity of monosodium glutamate. And we worked together with Kenneth, who I think had attended the third Ibro school, to create experiments that Sarah and Oliver, also pharmacy undergraduate students, could do to assess the toxicity of monosodium glutamate. Again, remember we had done exposure of the pink flies to hydrogen peroxide in the trend workshop. So we did again this similar experiment for them, and were able to see that when the flies were exposed to monosodium glutamate at different concentration, as low as 0.04% and as high as 5%, there was reduction in the scavenging activity of the fly extract. We also were able to see the catalase activity of the flies was reduced with increasing concentration of monosodium glutamate. Again, this was very simple because we just introduced monosodium glutamate in the food of the flies at that particular concentration, and then crushed the flies and were able to measure the catalase activity. We were also able to assess the lifespan of the flies that were exposed to monosodium glutamate, just putting the flies, feeding them, adding monosodium glutamate in their diet and assessing them for a period of time. And we could see the lifespan of the flies declined when they were exposed to monosodium glutamate compared to the control. We can see after 30 days, most of the flies that were exposed, in fact, all of the flies that were exposed to any form of monosodium glutamate had died, but we had some flies still alive from the control that was not exposed to monosodium glutamate. Again, we can also see the geotaxis. Again, interestingly, was slightly higher in the flies that were exposed to the flying up was slightly higher in the flies that were exposed to monosodium glutamate compared to the control. However, there was no significant difference. So you could be able to assess that monosodium glutamate exposure to the flies reduced their lifespan, but did not in the short term, but did not affect their locomotor activity. However, it did decrease their antioxidant activity. So these were very interesting insights that we're able to do to assess the effects of these compounds, fluoride and MSG, in the lab using very simple experiments and the fly. You can imagine if we had to do these similar experiments using rats, how long would have to feed the flies, to how long we had to feed the rats with MSG to be able to assess survival. How much we would have to, how much of the MSG, which was also expensive, we had to add to the fly food, to the mouse food or to the rat food to be able to see this kind of results. I also had a third student, John Mark. Now, I was able to work with John Mark. He was also a pharmacy student. I was able now to work with John Mark to move my results of the in vitro experiments into Drosophila melanogaster. So we now at this point we had a second round, and we had Imena coming to the lab to teach us now about to teach us more about. I think this experiment was the use of natural products, and she came with DPPH. And now we were able to expose Drosophila melanogaster to amaranth and compare it with ascorbic acid. And then we measured, after we gave them amaranth at different concentration for a period of 14 days. Sorry, I hope you have seen my slide. I have moved.
No, you've
No.
not moved.
You have
They are
moved.
not
Okay.
me.
To the left.
I've just
Yes,
remembered I didn't move. So okay, so this is the work we did with John Mark. This work was published in BMC Research Notes, and we're able to assess the in vitro DPPH scavenging activity of the amaranth, of the fly extract, okay, of the fly extract, after exposing it to either the amaranth leaves or ascorbic acid, which was our control. Now, the amaranths did not have as much antioxidant activity, as much DPPH scavenging activity as the fly extracts, the flies that were exposed to similar concentration of vitamin C. However, we did more antioxidant activity, and this was the in vitro DPPH scavenging activity of the amaranth leaf extract. This was the leaf. We had not fed it to the flies, just comparing the DPPH scavenging activity of the amaranth leaf and ascorbic acid. So DPPH is a free radical, okay, and it can be neutralized by antioxidants that are present in the extracts of the leaves of amaranth, and we compared with a standard antioxidant, vitamin C. In the next part of the experiment, now we fed the vitamin C and the amaranth leaf extract to the Drosophila flies. And then we were able to crush the flies and measure the antioxidant activity of these flies. Again, we still did the DPPH activity, inhibition of DPPH scavenging activity, and we can see the antioxidant activity of flies that were exposed to normal fluid, to amaranth, 0.05 milligram per mil, 1 milligram, and then ascorbic acid and ascorbic acid. Now, this was very interesting. In vitro, we could see ascorbic acid was a very strong antioxidant. But when we gave it to the flies and we measured the same experiment from the in vivo extracts, you can see the antioxidant activity of the extracts from flies that were fed amaranth compared to those that were fed vitamin C. It was even more interesting. The lower dose, 0.05 milligram per mil, had similar antioxidant activity as extracts from flies that were fed ascorbic acid at 0.05 milligram per meal or 1 milligram per meal. So this was very exciting to be able to demonstrate the in vivo activity of the amaranth leaf. All this time I had been doing amaranth outside in vitro, but now this was actually an in vivo and showing that when it's given to an organism, the amaranth had similar antioxidant activity to vitamin C. The next one we also did was to measure in vivo catalase activity because, of course, we could be saying, hey, all these antioxidant activities because for amaranth, but we also have antioxidant enzymes present. So we measured the catalase enzyme activity in flies that were exposed to amaranth as an exogenous antioxidant, and you could see, although there was not much significant difference from the normal, but there was slightly higher catalase activity, and the catalase activity was also higher in flies that were exposed to ascorbic acid. So again, I emphasize that this was very simple experiment. All we had to do was introduce the amaranth leaf as extract into the food of the flies, and were able to demonstrate in vivo antioxidant activity. Sorry. Okay, still with Mark, we were able to measure survival of the flies that were exposed to hydrogen peroxide over a period of time. Remember, we had seen that if you expose flies to hydrogen peroxide, their lifespan reduces. They die faster than flies that were not exposed to hydrogen peroxide. So in this case, we introduced them to hydrogen peroxide and flies that had already been fed on food that was rich in amaranth leaf extract for a period of, they had been fed in this amaranth leaf extract for 14 days, for 2 weeks. So we could see in the first slide, we can see flies that were exposed to normal food, were not exposed to anything. Even after 15 days, they continued. We didn't expose them to even hydrogen peroxide. So this was our control. Then we had flies that were exposed to normal food, and then they were exposed to hydrogen peroxide, and you could see their lifespan reduced. But we could see that were flies that were exposed to food that had either amaranth or ascorbic acid, they kind of survived a bit longer than the flies that were exposed to hydrogen peroxide without any exogenous antioxidant. And in graph B, we went on further to show how amaranth compared to the normal food. Okay, we can see these flies were able to live slightly longer. They went as far as 15 days, especially the ones that were exposed to 1 milligram 1 milligram per meal of the leaf extract in their food. They lived a bit longer. So this was very exciting, again, to be able to show the in vivo effect of antioxidants and compare it to the flies that were not exposed to any food. So I've spoken a lot. Maybe it's your time to tell me anything that. Okay. So from this session, maybe we can just take a moment and reflect on any experiment that can be easily done using Drosophila in a regular undergraduate lab. From what I've described and what I've explained in some of the experiments, which experiments can be done in a regular Drosophila undergraduate lab? Please use the same link. Any experiment you have seen that can be easily done with the equipment, using Drosophila with the equipment that we find in a regular undergraduate lab. I've talked about several experiments. Just go to the Mentimeter and mention any that you have seen or anything that you have seen you can be able to do using a regular undergraduate lab, not a sophisticated research lab. We're using the same code, the same window, the same QR code. I have shown you some of the experiment we've done with my students who are now doctors. So is there anything you are able to do yourself in your regular undergraduate lab? Yes, thank you so much. You're able to do antioxidant activity. We can we can see the antioxidant activity against free radicals like DPPH if you have it. Ferric, ion 2 to ion 3 is a free radical that you can easily use in the lab in the reducing power assay. You can be able to measure catalase activity. Okay, that is a good experiment. So you can be able to demonstrate in vivo antioxidant activity. Thank you so much. Again, yes, using a free radical like hydrogen peroxide. Okay, you can see how it affects the lifespan, how long the flies live, and this can be a very good. We used it to demonstrate the in vivo antioxidant activity of amaranth leaves. Okay, so in interest of time, finally, as I progressed, I was able now to do more experiments with my students. We even became a bit ambitious and tried to develop a diet for overnutrition. This was inspired with one of the slides that I can't remember who, either Drosophila or Trend, they taught us about how macronutrient content of diet can affect the lifespan, and they can affect either the lifespan and the reproductive activity of the fly. And they brought up the concept of choosing you can see when we had diet that was rich in sucrose, we had more increased production of eggs by the female, but less the lifespan reduced. So this started stimulating in my mind that what role does our diet have in some of the physiological activity? And so we tried to create a diet can cause overnutrition, because we do know overnutrition is associated with many problems that are currently facing us when you think about the double burden of malnutrition in Africa. So we tried to create a model that we could use for overnutrition. And we created two diets that were either rich in carbohydrate or rich in protein, varying in protein and carbohydrate ratio. And we exposed it to the flies, and we tried to see how the weight went. Interestingly, we found that diets that were rich in protein caused increase in weight. So these animals were these flies were heavier compared to flies that were exposed to diets that were rich in carbohydrates. But interestingly, when we look at markers of overnutrition, one of them is hyperglycemia. Flies that were fed a diet that was rich in carbohydrates had higher glucose, and their extracts had higher glucose concentration compared to the flies that we actually thought were heavier, that were obese or heavier in our case. When we looked at the triglyceride concentration, again, we find the flies that were exposed to high carbohydrate concentration, a diet that was rich in carbohydrate, had higher triglyceride concentration. So this was very interesting to us, because when you think about overnutrition, to find a model of obesity, not only are we looking for heavier weight, but you want to see biomarkers or biochemical markers that indicate overnutrition, like hyperglycemia, hypertriglyceridemia, and this we were able to demonstrate in the flies. We also, remember, this was just an undergraduate student, so we did not go very far with him, but also we were able to look at the survival rate, and we could see this diet that was associated with hyperglycemia and hypertriglyceridemia also had higher survival, but our high protein diet had lower survival. So this was very interesting to us, and you could think about, we used the explanation of high protein diet. The metabolism of protein is associated with high amounts of free radicals that could contribute to the lower lifespan. So again, this was a very simple experiment. just modulating, changing the amount of carbohydrate. Remember, preparation of food of the flies in the lab, we're using purified ingredients. And so we could be able to measure how much yeast we were adding, which was our source of protein, and how much sucrose and starch we were adding, which was our source of carbohydrate, to create diets and now see how these affect biochemical markers. And finally, I had, with time, I co-supervised a postgraduate student, Oscar, with Dr. Wampande. And now, because we had already had our interest in obesity, we created a model of obesity. And now we were trying to see Erythrina absinica, which is a plant that is commonly used in Uganda to manage many conditions because it is rich in antioxidants. And we do know many of the conditions we find in obesity, like cardiovascular conditions, hypercholesterolemia, are all associated with high amounts of free radicals. We tried to see if extracts of the bark of Erythrina absinica could be used to manage obesity. So in this case, we created obesity by adding coconut oil extract to the food that we are using. And we could see, and we used fenofibrate as a drug, as a positive control drug that is used in as an anti-obesity drug. And we compared it to the extract of Erythrina absinica. Okay, so we could see the survival. The first thing we assessed was the well-being of the flies, and we did this using survival, just measuring how long the flies lived. And we can see within 15 days, several of the flies that were exposed to that were first fed that high fat diet, the coconut food, had reduced lifespan. This is our positive control. We can see in 15 days the fly had died. However, when we exposed either this fly that was fed diet that had coconut food, and in addition to fenofibrate, or the extract from the erythina, we had rescued survival, and we had some of the flies surviving. We can see our negative control. This was just the fly that was fed in normal food. We did not have any mortality in 15 days. We went further ahead and compared how fit the flies were using the climbing assay. I've already told you about the climbing assay, negative geotaxis, and we could see flies that were not exposed to either the negative control that was not exposed to the coconut food had a very good climbing assay. However, flies that were exposed to coconut food, okay, the climbing assay was also a bit lower, but not very low. But when you expose them to the drug at different concentrations, the higher the concentration, the lower the climbing assay. The 25 millimeters and 50 nanometers of fenofibrate had lowered the climbing assay. Again, exposure to the extract from Erythrina absinica reduced the climbing assay. So even though the extract could have positive effects on lowering on obesity, it has effects on the fitness of the animal, possibly because of the taste or other effects that we are not able to access at that moment. We also checked in the geotaxis over time. We can see as we continued to expose the flies over a period of time, the flies were not even able to move. They were weaker as we increased the concentration. As we did to measure the in vivo antioxidant activity of these flies, we compared the in vivo antioxidant activity. Remember, this extract, literature had said, previous studies had shown it had high concentration of antioxidants, the terpenes and other alkaloids. So we exposed it to the fly in the food of the fly at concentration of 25%, 50%, and 75%, and compared with vitamin C. So you can see the in vivo antioxidant activity was not as high as vitamin C. So here we are comparing the reducing power. Here we are reducing iron 2 to iron 3. Again, we measure the cantalase activity of male flies who are exposed to both fenofibrates and the extracts. And we could see, again, the ones that were exposed to fenofibrate had slightly higher, slightly lower catalase activity. Remember, these flies were exposed to coconut food, coconut oil in their food to create obesity. So we could see the catalase activity was varied, was slightly different, but you can see the flies that were exposed to 25% of the extract had similar activity to the flies that were fed the normal diet without coconut. So it was interesting to see that as we have higher concentration of the extract, we could be having other negative effects like we have seen in the negative geotaxis. Please leave. Excuse me. As I come to the end of my presentation, we had other experiments that we did, but I can see we have gone past time. And so we also measured the triglyceride levels and the cholesterol levels. And generally, my take home from this talk is, yes, Drosophila can allow us to do elegant experiments that probably would cost a lot with other models. And as you can see, just their storage conditions, they allow us to be able to do many numbers of flies that in the experiments, they have a very short lifestyle, a short lifespan, and can allow us to do many remarkable experiments, not only for antioxidants, but also other diseases that we could be our area of interest. And we don't have to be limited in terms of funds and expenses to conduct research with Drosophila. I would like to give thanks, of course, to Firat for giving me the opportunity to present my work. Sayansi, Dros Africa, Trent, we are here because you came one day to visit us in Uganda and opened our minds to the possibilities of research. Of course, Kampala International University, my alma mater, and the IBR lab, and of course, Aga Khan for this opportunity and the time out of my regular schedule to be able to teach you. Thank you so much. Just last minute, did this session meet your expectations? Did this session meet your expectations? What you expected when you come to the talk? As we listen to this is my end of my presentation, and thank you for your time.
Thank you very much, Dr. Hellen, for the great presentation and session. It was indeed, for me, it really met my expectation. I don't need to use a Menti. Thank you for sharing your research journey. And it is an eye-opener for us to know how we can really move forward in real life. Like, since you have done it, it will be easier for us also to follow your procedures, to do more researches. I don't think I have a lot of comment on this. The slides were self-explanatory and you explained it very well. Let's just go straight to questions. Someone said I would like to learn how to use Drosophila melanogaster in a cancer research study. I don't know if you have some comment on this. In the chat box.
Oh, in the chat box. Yes,
Yes.
I believe it is being used in cancer research. It's not my area of research, so I may not have a lot of expertise. But I know if I just think about just on top of my head, if we have a drug that you think could have anti-cancer activity, you could probably start before you move on to higher models. You could start testing its antioxidant activity. Sorry, not antioxidant. It could be able to test its feasibility by if it has any toxicity in the fly first before you move to other models. Also, you can look, if you know about the cancer, what kind of mutation is available, has occurred. You can be able to see if there's a fly model that has been created with that particular mutations. And as something I might have not mentioned, the fly community is a very generous community. They do not hide and they are able to share their models. I think Professor Bolaji talked about the fly base and how you can be able to request for any flies. So just depending on the mutation that is available, you can see if there's a model of fly that has been created, and you can be able to use this model to test your drug, especially just on top of my head. But this is not my area of research. So yeah, thank you.
Okay, thank you for giving us your view. Maybe since you know many people, you can even help like link with someone who is doing something on cancer, I guess.
Yeah.
Also, the Dr. Tofik said that what form can corroboration can be done with, since your lab has done many things with using this. Is there some facilities that can help in for postgraduate experiments?
The experiments I was describing, we did them at the Institute of Biomedical Research at Kampala International University. I will have to confirm with colleagues because I left Kampala International University a few years back, although I've been in touch with my students. At the institution I am in, Aga Khan University currently, we've just started the School of Medicine, and Biochemistry is a new course here. But we've been talking with my supervisor, Professor Semiat, about the feasibility of using Drosophila to teach our students. I'm very passionate about being able to do the hands-on, especially since, for me, one of the groundbreaking and the mind-changing thing was be able to do something hands-on and actually see some of the concepts that were taught theoretically are physical. And because we are new, we're thinking about how we could bring in Drosophila and use it here as a not only for teaching but also for research. So in terms of facility for postgraduate research, currently at Aga Khan University, that is not available. But yes, but we are there to mentor and support in any way possible we can. We stand on the shoulders of giants in Africa and Trend. We can talk to some people. We know some people.
Okay. Very much. Also, there is a problem. There is someone who mentioned if you can use Drosophila in a drug discovery. Is there someone who did those
Yes,
experience?
yes. That's what I was describing was actually drug discovery in cancer. So yeah, especially now in Africa, there are so many compounds that probably could have been used traditionally. You know what we do here, and I think even Professor Bolaji talked about it. The feasibility is, especially with the models that have, for example, the one I told you, pink, any drug you can think about. For example, if it was a few years back, we have models of wound healing that could have used to do my ulcer. My ulcer started with Ficus natalensis. So probably we could have found a new drug that could be used for treating ulcer. These ideas were not available for me. But right now with Drosophila, it is possible. There are models for almost everything that you can use for drug discovery. Just think about your drug, what you want to use it for, find out if there's a model for it. And yeah, and when I was using, when we were using Drosophila, I know flies that were sent through the post office and you were able to come all the way to Uganda. Yeah, so yeah, it is possible to use it for drug discovery.
Okay. And what about malaria-based studies? Can those fly be useful in malaria field?
It's not my area of study, so I would not risk to say anything.
Okay.
So it's not my area. Probably it's an area we probably need to read into and see if it is used. And yeah,
Yeah, and we do our research
yes, and do our
and see
research.
if it
Yeah,
will work also.
yeah.
Yeah. Also, there is a question, if can we how can we get access of training from the Ibro lab? Do you
Yes.
have any idea of how?
From the Ibro lab, I came into contact with Ibro through Trend, and I know they're continuously sponsoring courses. So Trend in Africa is continuously sponsoring training courses across. I saw as recently as 2022. So I should share the link of Trend. They have a website where you can check and they've been teaching different courses in conjunction with IBRO, especially with people with interest in neuroscience. So yeah, it is possible to access IBRO training through Trend in Africa or directly IBRO. But yeah, I can share the link of Trend.
Please, thank you very much.
Yeah, I will share with.
Another, okay, thank you. Another one is a question related to what you presented.
Yes.
It mentions, please, did you probe further why the flies that were fed high protein diet had higher weight compared to those fed with carbohydrate diet and high lipid? Did you go further into that study?
No, we did not go. Remember, this was an undergraduate study. At undergraduate study, it's mainly just proof of concept, so we don't go further to elaborate. However, if you think about proteins, proteins are used for synthesis of muscle, cellular structures, and all that. So probably what you are seeing as heavier could be responsible for higher muscle mass. Probably it's also interesting to think the way we think about obesity in humans or in other animals, maybe like rats, who probably would not be what we see as obesity in flies. So in our model, we were able to see obesity in terms of biochemical markers, hyperglycemia and hypertriglyceridemia, which do not necessarily relate to high fat. So the metabolism, carbohydrate, especially lipid metabolism, is slightly different in Drosophila than in humans. So I think this high protein diet could contribute to heavier weight because of more muscle bulk or stuff like that. No, but we did not go further to try and establish what's responsible for this. But this is a good area that could go further and check out what contributed to the higher weight in the flies compared to those ones. Yeah.
Sure, it is another research area, and it goes
Yes.
with the other. You have answered the next question. It was also about the obesity and overweight.
Yeah.
So another question: how can we use Drosophila melanogaster in neurodegenerative diseases?
Ah, man, this taking me back. Oh, during my trend class. So yes, neurodegenerative. I spent a very interesting part in trend because, as I told you, trend are really interested in neuroscience, and a lot of work is being done with Alzheimer's, Parkinson's disease. Remember, I told you there are flies that are bang sensitive. Bang sensitive means that when you bang them, they convulse. And I know a colleague in Uganda who actually used the bang sensitive to study anti-epileptic drugs. Okay, so this is one of the neurodegenerative diseases. I have a colleague, one of the colleagues who has done a lot of work with Alzheimer's. I'm forgetting his name, but he also used Drosophila. So the cool thing is that in area of neurodegenerative diseases, we have models. So we have already well-established models for Alzheimer's. In fact, A beta defective flies, mutant flies are present. I know pink was a mutant for Parkinson's. One of the genes that have been found to contributing to Parkinson's is one that was used to make the pink fly. So in neurodegenerative diseases, I would say Drosophila will be an excellent model to use because we have flies that have been created that have similar mutations. And so you just need to identify which neurodegenerative disease you would like to study and find the fly and see how it can come to Rwanda.
Okay. After
Yeah.
seeing other questions, I think there is a other two which is different, because about brain cancer, they said that you didn't work on cancer things, and the memory. You have also mentioned. Maybe I'll bring these two remaining question. What is the easiest, if you have an idea of what is the easiest way one can use to secure a transgenic line of those flies in Nigeria?
Fly base. And you have Prof. Abolaji, you should discuss with him how he gets his flies into Nigeria. But as I told you, the fly community is very generous. Find someone who is using the fly, talk to them. In Uganda, we got flies coming by the post office in vials. And so I think it is possible, unless there are rules against transgenic flies. I know here in Kenya they are very sensitive about bringing new flies into the environment. So you just need to find the rules, the ethics surrounding it. But I think if you can get the fly, you can identify the fly and identify a community that is working with it, I believe they will be able to share with you.
Okay, thank
Yeah.
you. I hope the person got the answer.
Yeah.
Okay, can one use deep learning and machine learning in the research on Drosophila?
Oh, machine learning is a new field, not my expertise. So I will be safe and say I will not comment on that. I don't know much about deep learning and machine learning, although I would like to know. So yeah, I would not be
Yeah,
the perfect person to comment in that area. I can also see
still
comment as
undergoing
yes. It's a new
undergoing
area,
researches.
very exciting, yeah. And the things
Okay,
that they are doing there is mind blowing. So it will be a very good field to know. Yeah.
exactly. Okay, what major oxidative stress marker can I analyze? Maybe this is someone who is interested in
What major oxidative stress markers can I analyze for what? So there are so many oxidative stress markers, depending on the tissue, depending, I don't know what you want to do, but
May.
it's a very open question. But it's a very open question. So oxidative stress markers are varied, but it really depends on the question you want to answer. Maybe we can talk more. I will share my contacts. You can see,
Exactly.
yeah, what markers what exactly you'd like to study, and we can panel beat around and see what markers you could oxidative
Yeah.
stress markers would be ideal for your situation.
Maybe if you can share your email address, many
Yes, I
people
will
will be
share with you.
interested. Yeah.
Yes,
So the other one, like, can you extract RNA from the fries and use
of
them for?
course. Yes.
Okay.
Yes, you can extract RNA and DNA for your molecular studies if you need them. Yes.
Okay. So I think okay. There's the last one. Okay, thank you for sharing with us the impressive achievement. Please, I need to know if you had carried out in vivo comet essay on genotoxic substance such as heavy metals.
No, I have not done that impressive work yet, but I'm not putting it off for the future.
Okay.
Yeah.
Okay, I think we have a lot to really work on once, like, we get access to labs and the needed equipment. I think there's a lot of to work on.
Yeah.
Yeah. So if there's no other question or comments, can we move on? Since the time is not on our side, as you mentioned. Unless if you have something to add, Dr. Helen.
No, I thank you for the questions. I'm excited that it stimulated your mind as much as I've been stimulated by people. So if this is what and you're thinking about using Drosophila, please, please, please join the fly community. Yeah, it's a wonderful model. Thank you so much. Thank you for the time.
Thank you very much for the great presentation. I would like to welcome Nason to give us a vote of thanks.
Okay,
Mr.
hello
Nason.
everyone. Can you hear me?
Yes, please, go ahead.
Okay, I think I don't have much to say after this
I
wonderful
think there's a problem
presentation.
with his network.
Can you hear me?
Yes, yes.
Yes, go ahead.
Okay, so I don't have much to say after this presentation, but we have some few words to just thank the presenter. She has taken us to a high level in understanding how we can use Drosophila for research in our low income settings. because we struggle with finding money for research. But if we can have a model that can uh, help us do our research without spending a lot of money, so this will be a good thing. So thank you for your presentation. You have showed us a lot of things that we can do with Drosophila, and this is something that we can use. We can use for research. To the organizer and everyone, I know you work very hard to make this event take place. So I would like to thank you for your dedication and hard work to make this instrumental and smooth execution of this event. So your effort in ensuring that everything was in place and running efficiently has not gone unnoticed. For participants and everyone, I would like to thank you for these challenging questions because it shows that you have an interest in using Drosophila as a model to advance the research. To the institutions, especially RBC, University of Rwanda, and the others, uh, I would like also to thank you for your commitment to promoting research and development in challenging environment, uh, which we face in our everyday research. So the knowledge, everything that you shared with us is really valuable. And then we promise that we will do everything in our capacity to make sure that Drosophila models are brought in Rwanda and that we can use them to find a solution to helpful problems that we face. So thank you everyone for your time. I know that we have a busy schedules, but having you all in this presentation is something great. It shows that you are really committed, and then you want to bring this research at a level that will be appreciated by everyone to address the problems related to human health. So everyone, I thank you for your attention, and I hope we have these presentations frequently. We have as many as possible to look for different possibilities to use Drosophila as a good model to strengthening our fundamental research. So thank you again, and I hope we see you again very soon.
Thank you very much, Nason, for the great talk. I would like to invite the Boyga for the closing remarks.
Okay, thank you very much. On behalf of the entire team, I would like to thank Dr. Hellen Kinyi. She has actually motivated me a lot, and I believe it's the same with every other participant. She discussed the use of Drosophila melanogaster, particularly in low middle income countries with limited access to research funds. And what particularly amazes me was the simplicity of the solutions that have been deployed across the region. And we hope that we'll have more time to collaborate as it's anticipated. Hopefully, we meet soon in Aga Khan. I was there a few days ago, and we hope that we'll meet very soon physically. So, on behalf of the team, we acknowledge our speaker for the very detailed presentation and all the participants for their time. So, we hope to see you again on the 18th of September, 2024. Goodbye.
Thank you very much, Dr. Adeboyega. I think this marks the end of our today's session. Thank you, everyone, for your attendance, for your interactions during this session. See you again for the 6th webinar. Thank you very